Cool. All right. Welcome, everyone. Thanks for logging on tonight. I know Mimi's usually doing these, but you've got me tonight. A couple of things before we get started, and I introduce Kylie. Um, a couple events going on. We just got our speaker for next month from Stream to Sea. That date I have right here. It is April 10th. We'll be doing a virtual talk with Stream to See, the Reef Friendly sunscreen defog hair product brand. On St. Patrick's Day, we've got some giveaways and things, so hop on the boat, wear your green. The end of lobster season is the end of the month, so hop on the boat that last weekend. We're gonna do special lobster hunting drops Monday the 31st. I think Mimi might have some sort of lobster grill dinner get together of some sort to celebrate the end of the season, but she'll let us all know about that if it's in the works. Don't forget the Lionfish Derby, April 11th, and Reel It In on the 25th, the cleanup event. Other than that, this is Kylie. She's one of my friends from my graduate program from the University of Miami. I worked along with her with the Marine Megafauna Foundation during my program, and she also works with the Field School, which is a nonprofit associated with UM and the shark research there, and is doing her master's thesis on the guitarfish, which we get to sometimes maybe see. I know Doug's in here, and Doug was able to spot one off breakers one day. I saw one last year there. So she's going to talk to us about what we can do with any pictures of individuals that we see and tell us a little bit more about the guitarfish.
Cool. Thanks, Sydney. Super excited to be talking to you guys. I did my project on, or I guess I'm currently doing my project on Atlantic guitarfish, stress physiology, and individual photo identification. And I did this with Field School and the University of Miami's Shark Research and Conservation Program, and in collaboration with the Marine Megafauna Foundation. So I'm going to kind of go through my research in blood physiology and photo ID, and then I'll go through how to take a proper ID photo and then where to send it in if you guys ever come across one while you're diving. We don't have that many people here today, so feel free to stop me whenever if you have a question. And then there'll also be time for questions at the end. But feel free to unmute yourselves and ask questions as I go. I know that some of these graphs look a little scary, so you can definitely stop me at any time. It's going to take a minute to, doesn't want to go. Wow. Okay. So like Sydney said, my name is Kylie Powers. I am originally from Missouri City, Texas, and I did my undergrad at Texas A&M, where I got my BS in environmental geoscience and a minor in oceanography. And then I headed to the University of Miami, where I currently am for grad school, and I'm doing my MS in marine ecosystems and society. And I'm currently a field school and SRC intern and a dive master in training. So before we get started on my research, just a little bit of background on Atlantic guitarfish. They are benthic shark-like batoids, which is another fancy word for ray, and they are found in the West Atlantic as far as North Carolina, stretching into the Gulf of Mexico and extending into the coastal waters of Brazil. They live in shallow sand, hard bottom, and patch reef habitats in less than 30 meters of water, and have a maximum recorded length of 78 centimeters, although the lengths we have been recording them at here are more commonly between 50 to 60 centimeters. And additionally, they are severely understudied with less than 10 published papers on them, primarily over their reproduction and feeding mechanisms. So Atlantic guitarfish are actually the only guitarfish found on the eastern side of the US. And while there are no range-wide population estimates, increased trawling from fisheries and overall decreases in fisheries catch of elasmobranchs, which is another fancy word for our cartilaginous fish, such as sharks and rays, has led assessments to estimate around a 30 to 49% decrease in Atlantic guitarfish populations over the past 18 years. And most of what we know on their biology and ecology actually comes from fisheries bycatch data. While Atlantic guitarfish are listed as vulnerable by the IUCN 2020 assessment, they are part of the rhinopristiform order, which includes guitarfish, wedgefish, banjo rays, giant guitarfish, and one that you guys probably know more commonly, the sawfish. And so these species are representative of a group that is extremely overfished and threatened. Therefore, research into Atlantic guitarfish will not only provide us data on a severely understudied species, it also provides us a better opportunity to research guitarfish populations that are in better shape, such as our Atlantic guitarfish, which can provide important data on this rhinopristiforms group as a whole. So some threats that they are currently facing, guitarfish face several threats, including being either targeted or brought up as bycatch in commercial handline and gillnet fisheries. And when targeted, they're typically utilized for their meat, as their fins are too small to be of value in the fin trade. And while it is unknown how much they interact with trawls on the west coast of Florida, in Mexico, guitarfish were the third most abundant bycatch species in shrimp trawl fisheries. Guitarfish are also threatened by habitat loss and degradation from coastal development and oil exploration that is occurring throughout much of their range. And habitat loss does include the study area of Palm Beach County, Florida, as human populations and demand for urban coastal areas has increased dramatically within the past 50 years. The image on the right, if anybody has ever visited Boynton Beach, this is the sandpipe at Boynton Beach Inlet, which is pumping sand from the north side of the inlet directly onto guitarfish bottom, a hard bottom habitat, which is just one of the ways that guitarfish are being threatened by habitat loss in Palm Beach County. Additionally, it is kind of funny to point out that the IUCN distribution map says that guitarfish are not present in Palm Beach County. However, our surveys do tell us otherwise. So in order to better understand this severely understudied species, I used two different approaches. The first one was to establish a physiological baseline for Atlantic guitarfish by using whole blood and plasma. And then part two was validating the use of the photo identification software i3S Spot as a method for studying Atlantic guitarfish movement ecology. So moving into blood physiology, I'm not going to really get into the process and reaction to a stress event, but we did measure lactate, which is the enzyme that makes you sore after a workout or other physically demanding activity. And we wanted to understand how capture or a stress event affects the lactate of guitarfish. So this study will be the first study to look at the blood physiology of guitarfish. And while we don't really know that much about them, we can make some inferences on other benthic ray physiology studies, such as those done on long-nosed stingrays and Atlantic guitarfish, which suggests that benthic batoids, which benthic is just another fancy word for an animal that likes to utilize that bottom sandy area. They have lower metabolic rates, lower lactate levels, and a greater resiliency to capture stress. The most notable of these studies showed this correlation with small tooth sawfish. So even though we think Atlantic guitarfish will be similar, we should understand their specific physiology as lactate levels do still fluctuate between these benthic species, and it will have important implications for their survival rates and sublethal capture effects because, like I said earlier, they're caught as bycatch quite frequently. So we did our study in Atlantic Florida, which provides critical information about Atlantic guitarfish ecology and biology, as they are largely protected from commercial trawling and capture in this part of their range, although they are still affected by that coastal development. Sample sites were chosen based on previous sightings of guitarfish during manta surveys for the Florida Manta Project that's run by Jessica Pate. And the study sites for blood physiology included breakers, which you guys mentioned before the presentation that you guys have seen guitarfish at breakers, and also Lantana, Boynton Beach Inlet, and Deerfield Beach. The first sites are in Palm Beach County, and then Deerfield Beach is actually located in Broward County. And the bottom habitat at these sites is mostly hard bottom and sand with an average depth of about 10 to 20 feet. So field days were either run from shore or in collaboration with the Florida Manta Project on their small boat. And snorkelers used roving diver technique, and we generally swam north and south along the hard bottom, usually focusing on the eastern edges where the hard bottom met sand. And we collected blood samples from October of 2023 to August of 2024. You can see in this picture here, there are 4 snorkelers in the water, and that's how guitarfish were located. And then they were captured by a cast net and then placed into a shallow seawater filled bin. This is a video of the capture process. So I'm dropping a cast net down over the guitarfish, and then once I have it in place, I can go ahead and grab the guitarfish and bring it up to the bin. Please don't do this. Actually, we have special permits for this. So please don't go do this if you're just snorkeling out in West Palm by yourself. So after it was brought to the bin, we took a blood sample via caudal venipuncture, which is basically just using the vein that runs underneath the spine to get that blood sample. And we didn't take any more than 3 milliliters of blood. And then we got our lactate value using a point of care device, which I'm going to go into further detail the next slide. Once we had our sample, we stored our whole blood on ice, and it was taken back to the lab where we then spun it down for plasma and collected that plasma and stored it in a negative 20-degree freezer. And then the plasma was later used to obtain a laboratory lactate value. So these are our POC or point of care devices. Traditionally, whole blood and plasma lactate are measured using laboratory assays in a lab using enzyme-based spectrophotometry. Really fancy word. It's just the analysis we use to run all of this in the lab. However, these analytical techniques are not readily accessible in remote areas or field settings and can be expensive, which has led to the use of these point of care devices. And we obtained our lactate value using the Lactate Pro 2 POC device. This has been originally used and extensively tested in human and veterinary medicine. And POC devices can provide nearly immediate physiological results, which has led to their increasing popularity in blood physiology studies. And the 2 main advantages of using this POC device are the reduced cost compared to laboratory methods and their increased accessibility. So POC devices such as our Lactate Pro 2 pictured here provide a much cheaper alternative to traditional laboratory assays. However, they do require species-specific validation. So I cannot, I'll kind of go into this later, but I come up with this equation on the next slide, and we cannot use that across species. So I'm going to show a lot of graphs, but we're going to walk through it nice and slow. These do look a little crazy, especially the next slide. But essentially, we took a blood draw and obtained a field lactate value using the device that you saw pictured on the previous slide. And then the rest of the blood was spun down in a centrifuge to collect plasma, which was run in the lab to obtain a secondary laboratory lactate value. So the field value and the lab value were compared to see how they differed from each other. So what we're looking at is a visual representation comparing the field device from the laboratory assay run, and we were able to come up with that correction equation you see in the right-hand corner, y equals 0.91x plus 1.93. It's also showing that in this case, we're seeing that the lactate probe field device is actually consistently and predictably overestimating our lactate concentrations compared to the lactate we were running in the lab. So these results are really important when thinking about its application in remote or field settings, with researchers being able to use this equation. So they can now use this equation in field settings where laboratory assays are not available or they're too expensive. So we can kind of think back to our example of guitarfish being brought up as bycatch in Mexico. So if I wanted to go out there and collect blood samples from these guitarfish being brought up as bycatch on a trawl boat, I could use this correction equation and plug in my lactate value that I obtained from my field device, plug it in my correction equation, and instantly have my laboratory assay value without the need to actually run the laboratory assay because they're really expensive. And I'm in Mexico on a boat. I probably don't have access to that assay anyway. So these are a lot of graphs, but we do know that different variables are known to influence lactate levels. So basically, this is just showing that we compared our lactate levels to sea surface temperature, total length, hematocrit, which is the measure of whole blood cells, or sorry, red blood cells to whole blood. And we know this is important because our red blood cells are what carry oxygen. And then also the time from capture to the actual blood draw itself. And out of all of these variables, sea surface temperature was the only one that correlated significantly with the lactate levels we were seeing, both in our field device and in the lab. So kind of looking at this trend more closely, we can see that both the field device, which is on the left, and the laboratory assay, which is on the right, correlated with sea surface temperature. We had a range of about 22.8 to 30.4 degrees Celsius in our sea surface temperature during the study. And for every 1 degree Celsius increase in our sea surface temperature, there was a 0.45 millimoles per liter, which is what lactate is measured in, increase in lactate for our lactate that we ran in the lab. And then there was a 0.26 millimoles per liter increase for every 1 degree increase in sea surface temperature for what we were seeing on our field device. So these results essentially better help us understand how Atlantic guitarfish will be affected by rising water temperatures due to global warming. While the present study suggests Atlantic guitarfish do display a high degree of resilience to physiological disruptions, this could decrease as ocean temperatures continue to rise, especially where populations are subjected to trawl pressures and habitat loss, like we're seeing in Palm Beach County. I know the blood physiology stuff is kind of hard to digest sometimes, but we're going to move into the second part of the study, which is photo ID, which I think is a little bit easier to kind of wrap your head around. There are no previous photo ID studies on guitarfish, so this is the first one. It's more commonly used on other elasmobranch species like eagle rays and whale sharks to study their long-scale migration, small-scale movement, and sight fidelity. But now it's becoming a more common tool for other species like small spotted cat sharks and sand tiger sharks, and it has been successful to study that movement ecology. So if validation of this software is successful, our photo ID would be extremely valuable for long-term tracking, as the ID software is lower cost than physical tags. It's actually free to download and use, and it would eliminate the common physical tagging limitations such as shedding, fouling, and failure. So these are our study sites. They're pretty much the exact same as the blood physiology, except we added Oceanfront Park, which is seen right there. And then we also had two photos taken from Phipps Ocean Park, one at Coral Cove and one at Datura Ave Shipwreck Snorkel Trail. And our ID shots were taken from July 2019 to October of 2024. At least for the data I'm showing you now, we have added more since then. And again, bottom habitat was the same with an average depth of 10 to 20 feet. So, ID shots were taken opportunistically, either out of the water as part of my blood collection workups, in the water during small boat days, as part of the Florida Manta Project surveys, or from shore-based snorkel surveys. We took all of our ID shots using a waterproof camera or a phone, and the rostrum or the snout, which is pictured here, was chosen as the comparison region due to the guitarfish body usually being buried under sand. And because I thought that the rostrum was easier to distinguish visually between individuals compared to other regions on the body. So this is a screenshot of what a photo looks like once it's kind of been annotated or marked up in the database. I chose the tip of the rostrum, the left spiracle, and the right spiracle as reference points, and these can be seen in the blue lettering. And then the area between these reference points is the comparison region, which I marked with these red lines. And that was for all photos inputted into the database. And only circled spots within these three chosen reference points were used for comparison and identification, and that can be seen as the yellow circles in the picture. And we can circle a maximum of 150 spots within these reference points. So photos were run through the photo ID software i3S Spot to determine if we could, in fact, use these spots to individually ID each photo. After each photo was run through the database, it was visually inspected against the top 10 most likely matches, which I'll show you kind of a screen of what that looks like on the next slide. And this verified that the algorithm was working and the first choice that the software provided was correct. However, we're hoping that this step will not be required in the future under a certain match score, which I'll show on the next slide again, once the database is verified for use on the species. So this is what the photo looks like once it's run through. Once the image is run through the database, a similarity value is given to the top 10 most likely matches, which can be seen here on the right. And a lower value indicates a better match between two images. Newly identified individuals are given a unique number to distinguish them. And if a match is found, the individual was marked with the same identifying number as its match. And we also took notes on location, date, sex, disc width, total length, and other unique features, and included them in the metadata for each picture. And I actually needed 200 individuals to verify this database. So again, this is what the software looks like once an image is run through. The original image is on the left, and then the top 10 most likely matches are on the right. If an image was thought to be the same, they can be pulled up. Oh, they can be pulled up side by side, and the regions of each ID shot can be zoomed in on further for comparison. The example here is a match, so the image would be included in the database using this button here, and marked with the same unique ID number as its match. If this image was a new individual, it would be given a new unique identifying number and then added to the database as a new individual using this button. So I'm going to talk about what we've been able to come up with so far for the photo ID software. We have, or at the time I was running the photos, so this again went just to October of 2024. We had run 283 images through the database. 200 of these were individuals, and then we had 83 matches. Of the 83 matches that I3S Spot identified, 10.4% were not correctly identified as the best possible match. So because of this, we did this analysis to evaluate the reliability of the software to actually accurately identify individuals. So we did an ROC curve to test the software's effectiveness by comparing the rate of true positives. So this was the number of individuals that once they were matched were ranked as number one, to false positives, which were images that had a match but were ranked not number one, so like 2, 3, 4, all the way down the line. And overall accuracy was measured using this AUC, or area under the curve. And all you really need to know about that is that the closer the value is to 1, the software is accurately identifying a match. So with an AUC of 0.93, we were able to show that the software demonstrated a high level of accuracy in its ability to successfully identify individuals using that rostrum spot pattern in the database. So this is showing the accuracy of the software again. So like I said earlier, I3S gives us a match score. So photos that were incorrectly matched were considered false positive matches. And then photos that were correctly matched were considered true positive matches. And we ran their corresponding match scores to get this violin plot. Violin plots are used to show the distribution of the data. So the wider sections of the plot indicate areas where more data points are concentrated. And this analysis calculated a threshold match score of 3.39, below which there are actually 0% chance of false positive detections and an 86.7% true positive detection rate, which means that below a match score of 3.39, the software will correctly match an image 86.7% of the time if it has a match. So these results establish that photo ID is effective at identification, and it shows that Atlantic guitarfish have a high spot pattern variation. The software was also validated on the use for Atlantic guitarfish, with 200 individuals being identified. So we can now use this as a tool for longer term tracking compared to physical tags, which makes studying their movement ecology a lot more accessible and eliminating limitations such as expense and tag shedding or failing. This is a fun little graph of 8 individuals and where they traveled. It doesn't really accurately represent the movement of guitarfish because we don't know where they traveled between their sightings, but it's a good way to visualize the distance traveled between each sighting. The furthest observed travel distance of an individual, which is Guitarfish 67, which is this purple dot that's going to go way up north, was approximately 23 kilometers. And the individual was seen twice at Oceanfront Park and then once at Breakers between a 398-day period. We also had movements of Guitarfish 14, 35, and 36, which were observed between Lantana and Boynton Beach Inlet, with a range of 292 to 436 days between those sightings. And you can see other movements of guitarfish on this map, but these movements were relatively short distances. So the days between first and last re-sightings ranged from only 4 all the way to 841 days. So this guitarfish that you can see on the screen was the guitarfish that was spotted 841 days apart. The image on the left is the first ID image taken in the water during snorkel survey, and then the image on the right is the most recent ID shot that was taken during a blood workup on shore. And then I also included a screenshot of the marked up image next to its corresponding original ID shot. So while more spots are visible due to the higher image quality of the ID shot on the right, I3S still provided a match score of 4.92, which is relatively low, and ranked it number one, correctly identifying this individual, which shows that spots are staying relatively consistent across the adult life stage, at least for a few years. And the software can accurately match individuals even when image quality is not great. So some future directions, we saw high site fidelity through the photo ID and had a 22.5% resighting rate, which is similar to resighting rates of other studies using photo ID on shark and ray species. However, surveys were not conducted outside of the study locations to evaluate if long distance movements were being made. We actually have another project going on using acoustic telemetry. So guitarfish get a small surgery. They get a little acoustic tag implanted into their body cavity, and then we're actually able to look at more fine scale movement. We have a lot of different acoustic receivers going from West Palm to South Miami, and if a guitarfish swims within a certain distance of those receivers, we can pick it up and we know better or have a better idea of where they're moving. So our preliminary acoustic tagging data on Atlantic guitarfish suggests that they are capable of making these long distance movements, with one tagged female making a return migration of 160 kilometers to its original tagging location over a 15-day period. So we really need more data to fully understand the habitat use and site fidelity of this species, as this knowledge could provide better insight into management practices where Atlantic guitarfish are threatened by those trawl fisheries and coastal development. We also know that spot patterns were shown to remain stable in adult guitarfish, but it is not known how spot patterns change across their life history. Researchers, or we actually have started a captive database to better understand the validity of the I3S software throughout their life history. So we're actually using captive guitarfish where we know each individual, and we're doing month by month photos and running them through the database to see if those spots remain consistent throughout their entire life. And this study also established photo ID protocols for future spatial analyses that can be applied to other guitarfish populations, where insights into their habitat use and movement ecology will be beneficial in creating better management strategies, with the potential for citizen science opportunities, such as those already in place for whale shark and manta rays. And oh my gosh, good thing we're talking about this, because now we have this really, really cool citizen science project that you guys can get involved in, especially if you're seeing them out on your dives. So you spotted a guitarfish. How do you get involved with the research? I'm so glad you asked. Um, I know that we've shown a lot of pictures on a slideshow, but I'm just going to go through real quickly how to ID a guitarfish. They are a small ray species, around 20 inches long. They have a shovel-like nose, white spots covering the majority of the top of its body. They're kind of brown, green, tan in color, and they have two dorsal fins set far back on the tail right here. And they can usually be buried, which is that top picture, or resting on the sand or hard bottom, like in the bottom picture. So when you're taking an ID shot, please, please, please do not disturb them. If they're buried, do not disturb them to get an ID shot. They often are buried. They really like to hide. But please still send in your sighting reports, further our knowledge on their habitat distribution and migrations. So if you want to take a picture, just don't disturb them, and then let us know where you saw it. When you saw it, it will still give us a lot of information, as we don't really know anything about them. If the guitarfish is not buried, I would recommend recording a video, because there's a less chance of the guitarfish swimming off while you're trying to get in the perfect angle for your shot. And also decreases the likelihood of a blurry photo. because you can pull multiple stills off of a video. So that gives you a lot more chances of being able to have a usable photo ID shot. You can focus on the face or the rostrum. This is the ID area. So you don't have to take a picture of the whole guitarfish. You can if you would like, but that is our designated ID area. So I have to crop the image anyway, just to get that face so that I don't have to zoom in as much. And the higher quality of the photo, the better. You can see from our photo ID section earlier that I crop the photo and then I actually have to zoom in extremely close to accurately circle all those spots. Yes, I do circle every spot by hand. Takes me a while. So the grainier the photo, the less likely it is that I'll be able to use it for an ID shot. So how to report your sighting. So you have your photo, you have a good clear photo. You can send that into Florida at marinemegafauna.org with the photo, the date and time of your sighting, the location of your sighting, and then also any other distinguishing characteristics. Sometimes they'll have scars on their face or they could be missing a fin. So we would love to know that information if you see it, because that can also help us ID these animals as well. That is all I have. I will take questions, and if you have any other questions, you can also email me at that email, kpowershsc@gmail.com. And if you would like to listen to more Guitarfish research, I do have a podcast on the Fisheries Podcast, which is available on Spotify, where I talk a lot more in depth about this and all the other stuff I'm doing at Miami, if anybody is interested in that as well. But that's what I have, if there are questions.
So I'm one of those lucky people. I've seen one at Ocean Reef Park, just snorkeling, and then one up in Jupiter near the um, Loran Tower dive. We weren't there, and I saw one, and then just one a week or so ago. Uh, that was the backside of Breakers, out on the outer reef, and much the places you've kind of mentioned as their common habitat.
Mm-hmm.
So no, really neat to see some work on this. I'll
Yeah.
definitely start trying to log things.
Yeah. I'm excited to see how much we can learn from this photo ID stuff as we start to integrate our citizen science now that I've validated that software. So I think we'll start being able to get a really good sense of where they're moving.
Have there been any attempts of doing something, like the eDNA, the environmental DNA, so finding a marker for these guys to estimate presence or prevalence?
We do take genetic samples. We take a fin clip, but we have not started a project with that yet. Um, we just haven't found the funding to kind of get that started. But maybe one day.
PhD.
Yeah.
No, really neat. Thanks, and make sure that we get some of the other links. I think I grabbed your email at least, but yeah, I'd love to get some photos and send those out.
Yeah, please, whenever you guys can. There is the all the other info is on Facebook as well. So if you look up Marine Megafauna Foundation on Facebook, all the info on where to send it um is on Facebook. It has all of this stuff as well, and then the email too.
I
Hey,
think I
Kylie.
just saw you and Jess post that the website is live with more details about y'all's research right now as well. I can also put that in the Kyalami connection. I'll grab a grouping of links from Kylie and post that as a follow-up with the recording link for this so people can have quicker access to that and rewatch this if they're interested.
Yeah, I'll put I think someone was asking about the podcast. I can put the podcast link. I can give that to Sydney as well as the new link to the website, which just went live today. So that will have all the info in it as well.
Hey Kylie, I may have missed it, but what method do you use for tracking the guitar fish on those plotting that you were showing them going up and down the coast?
The map, right?
Like, do you tag them? Do you tag them at all when you do your testing and of them, or how do you know that tracking?
So this map is based on the photo IDs. Uh, so I took where they were sighted once, and then I took where they were sighted again, and that's where those lines are going. So we don't really know where they're going between the sightings, because it's not an actual physical tag. Um, but it kind of gives us a sense of how far they're moving, if that makes sense.
It does. Thank you.
Yeah.
How do you think they would respond to some kind of tagging or satellite tracking? Think it would do damage to them?
Yeah, we actually went back and forth on this one. So I work for the Shark Research and Conservation Program here at Miami, and so we tag all of the shark species here that you can find in Biscayne Bay. And we use what's called a spaghetti tag. So it's essentially just like a little plastic. I wish I had one because I'm actually in our lab right now, but there's none in here. It's about this long. And it basically looks like what you would think a piece of spaghetti would look like. That's the thickness of it, but shorter. And we'll put that underneath their first dorsal fin. But with these guys, their fins are really thin, so they don't have enough tissue for that to hold. We think it would be really damaging, and then would end up falling out within probably a few hours. Um, so we did talk about that. We are doing a tagging study. My partner in the project, Jenny, is doing an acoustic tagging study where the tag is actually implanted into their body, and that can ping off of our I don't even know how many receivers we have out now between West Palm and South Miami. So if they swim past one of those, it detects it, and then we can go download that data and really see where they're moving. So hopefully one day we can combine the photo ID with this acoustic data to get a really, really good sense of how they're moving. Anybody have any other
Oh,
questions for
hi,
me?
hi, Kylie. It's
Hi.
Papa. What is the purpose of the fish burying themselves other than camouflage?
Mm. It's mainly just to hide and rest. Um, Predator avoidance. That's the main thing.
So they don't feed all the time?
No, no. As they're feeding, when we find them foraging, they're eating stuff in the sand and on the hard bottom, so they'll be out and swimming and kind of sifting through that like a ray would. Like I'm sure you guys have seen videos of stingrays and they blow up all the sand while they're searching for food. Guitarfish don't do it quite so dramatically with all the sand, but they do use that same technique where they're going over the sand. They're kind of sifting through it, and then they'll move to a new spot, sift through it. That's how they forage.
Are there any quick sexual dimorphic things? So like claspers or anything we can kind of sight in on to give more information if we see one or
Oh yeah, good question. So yes, the males do have claspers. Um, if anybody doesn't know what a clasper is, it's a modified calcified pelvic fin. It's basically an external sexual reproduction organ on males. So I would say the only thing with that is sometimes you cannot see them because they're underneath the body. So if they're swimming or they're resting, sometimes they'll stick out, but depending on the size and maturity of the male guitarfish, you may not see them. Um, so if you see them, please let us know that it's a male, because that is good info. But otherwise, if we don't flip the guitarfish over and check, we don't know for sure. Um, I think, and I tend to see that the females are larger than the males, but if you haven't looked at a ton of guitarfish, you probably wouldn't be able to tell just by looking at it. So I would use those claspers as the best indication. Otherwise, they look the exact same.
Hi, Kylie.
Hi, Nana.
Hi. I'm wondering, is much known about other aspects of their lives, such as how they reproduce and lifespan and yeah.
Nope.
Okay.
There's a few studies on reproduction that have looked at litter size, I believe, but that was done back in the 90s, and there hasn't been anything new about that. From my research and just observations, the females seem to reproduce actually around this time, around spring, February to May, I would say, is when we're seeing pregnant females. Um, but we don't know where they reproduce. We don't know when they reproduce. We don't know how often they reproduce.
Is it a
Uh,
live birth?
um, I believe so, yes.
Okay. Thanks.
Yeah. But we actually have picked up a pregnant guitarfish on our photo ID that was pregnant and then was not pregnant, which was really cool to see.
Awesome. Thank you, Kylie, so much
Of course.
for chatting with us. Yeah, like I said, I'll post any resources, links, that podcast I guess you were talking about, where you can submit your sightings. And yeah, I think Kylie's gonna come diving this week, so that's gonna be fun. Um,
Yeah.
yeah, but you guys know if you have any questions, you can reach out through Kyalami Connection or to me directly. And thank you guys so much for logging on tonight.
Thanks, everybody.
thank you. Bye.